Hi there, today I'm going to be running through PCR, a research technique I used during my summer research project. PCR, the polymerase chain reaction technique, was developed by Carrie Mullis in 1983 and is now one of the most powerful tools in molecular biology. PCR essentially acts as a copying machine for DNA, used to amplify and detect specific DNA molecules. First, RNA must be extracted. But as PCR does not work with RNA, the RNA must be converted into DNA using the enzyme reverse transcriptase. This DNA is called cDNA, complementary DNA, which is complementary to the previous RNA strand. PCR takes place in a PCR machine, also known as a thermocycler, used to heat and cool the DNA strands. Typically 30 cycles are performed. However, PCR machines can be programmed to be used for lots of different cycling parameters. The neon green F label in each image throughout the video represents fluorescence. I will come back to this in a minute, but first I would like to break down the 3 key steps in PCR. The first step involves the heating of DNA to break the bonds between the two strands. This helps separate the strands, and this is called denaturation. In the second step, the temperature is lowered to allow for primers specific to your chosen target DNA to bind. This is called annealing. In the third step, the temperature increases again and the enzyme DNA polymerase adds nucleotides, which are essentially building blocks, to the end of primers using original DNA strand as a template. This is called elongation. The polymerase used in this reaction is also known as Taq polymerase. It is very stable in hot conditions, surviving temperatures between 50 and 80 degrees Celsius. PCR continues cycling between these 3 steps, doubling the amount of DNA each time. However, PCR does not have unlimited nucleotides, therefore it cannot create an exponential amount of DNA, so the PCR amplification curve plateaus off. This also means that if you were comparing multiple samples, they would have the same amount of DNA at the end. Despite this, by comparing curves, you can work out the amount of template DNA you started with, gaining quantitative results from the curves. Real-time PCR monitors the amount of DNA in the PCR tube in real time after every cycle. As I said, the neon green F label in each image throughout the video represents fluorescence. This is essential in real-time PCR, where a probe is used which gives off fluorescence when a new DNA molecule is formed. In real-time PCR, the increase in fluorescence signal is monitored whilst amplification occurs. Once the fluorescence signal exceeds a certain threshold, you know that the fluorescence signal is significant. This generates a cycle threshold, CT value, giving a value for the number of cycles required for the fluorescent signal to exceed the threshold value. This is marked on the diagram by the red dotted line. Running PCR tests generate a lot of results in a relatively short amount of time, therefore being an extremely useful research technique. And so that's all for today. I hope you have found this to be an informative video.